basically today I'm going to be talking about some of the risks associated with establishing the contribution of phytochemicals to the human metabolome. I'm going to use polyphenols and berries as a case study, but the same would apply to all foods. And just to start to work the audience, how many people know what mass spec is? Show of hands. Okay, leave your hands up if you've used a mass spec. Okay, so we're pretty good. We're about 50-50. So this is what we're up against. So we have a diversity of phytochemicals in the diet. You've probably all seen this chart. We've got our carotenoids, our phenolics, alkaloids, nitrogen-containing compounds, and organosulfur compounds. I'm going to be focusing on this subclass. So we have our phenolic acids, things like hydroxybenzoic acids and cinnamics. We got our flavonoids, which most of us are familiar with the multiple classes that you see down here. And then we've got the tannins, which are polymers based on flavonoids that can also be linked to phenolics. So we've got a vast diversity of phytochemicals. So why am I focusing on this kind of class here? And this is a slide from Mario. Is basically there's a vast abundance of these things in our diet, so we need to really consider them in metabolomics research. If we consider both the monomers and the polymers in commonly consumed foods like cocoa or tea or apples, it's conceivable to consume a gram of these compounds within a day. Furthermore, when you consider the low molecular weight phenolics in coffee, say chlorogenic acids, or in berries, again, we could easily consume a gram of these things a day. So we're looking at 2+ grams of these phytochemicals in our diets contributing to our human metabolome. So what makes this even more complicated is we have phase 2 enzymes that metabolize all these compounds from plants, but also we have this microbiome to consider, where we have microbial catabolism, taking these compounds, breaking them down into benzoic acids, phenylacetic acids, cinnamic acids. And if we take just this kind of underground subway map and go through the metabolism of just one of these polyphenols in our diet. This is cyanidin 3-glucoside. It's converted to cyanidin. Then we have it early breakdown product, a caffeic acid, to a dihydroxyphenylpropanoic, dihydroxyphenylacetic. This caffeic can be methylated, forming isoferrulic and ferulic. These compounds here can go on to form hydroxyphenylpropanoic acids and hydroxyphenylacetic and mandylic acids. And again, this is all from one compound. We further could follow this train down to dihydroxyferulics, methoxyhydroxyphenylacetics. And at this point, the next set of breakdown products, basically the structure is getting smaller and smaller. We get the formation of various benzoic acids. The terminal breakdown products, as far as microbes, before ring fission, I should mention before that, all of these compounds, when absorbed, can also undergo phase 2 metabolism. So methylation, glucuronidation, and sulfation, forming human microbial hybrid metabolites, essentially. So the microbes will eventually break these things down before ring fission to simple phenols like phenol, catechol, resorcinol, and pyrogallol, which are essentially, this chain is completely removed at that point. And then the human's ultimate route of excretion or terminal metabolite would be hippuric acid and hydroxyhippuric acids. And that involves glycination. So you can see one phytochemical in one subclass has a very complex metabolism. Now, we also need to consider our absorption, distribution, metabolism, and elimination of these compounds, which again is further complicated by the microbiome. So when you consume a compound, it's obviously in high abundance in the upper intestine. We're able to absorb this compound, which gets conjugated both in the small intestine and the liver. Again, then it would circulate in the blood and be excreted primarily through the kidneys. But as these compounds sit in the intestine and move further and further and further down, they encounter higher densities of microbes within our microbiome, causing all of these catabolic reactions, breaking these structures down, and then these structures too undergo phase 2 metabolism in the intestine and liver before entering into the systemic circulation. Now, obviously, this is going to involve differences between individuals as a result of gastric emptying and intestinal transit, which is going to affect kinetics significantly. So what we're seeing with kinetics is we have this early absorption and metabolism, so we get these little spikes early on, but then these microbial metabolites, if we can consider them more of a later phase absorption and metabolism, are showing this massive peak with very long elimination kinetics over 48 hours. So we have a first pass effect, which is entering the systemic circulation and going through systemic hepatic recirculation, essentially. And then this later phase, which is going through the same process. And what that's doing is that's meaning that metabolites in one pool can cross-jump and become a metabolite in another pool. And that's making it very hard to model kinetically. And in fact, we have no modeling software to model the contribution of the intestinal microbiome to kinetics at this point. So then how do we take all this complexity and work out the contribution of phytochemicals to the human metabolome? So we started by developing a database, essentially. So first we want to know, okay, what's in the food? So let's take berries as an example. We go to Phenol Explorer, the USDA database, various online databases, and try to work out what's the composition that's going into the system. Now, these databases tell us that berries in general are very similar. So if they're red or blue, they're high in anthocyanins. They also have a number of other compounds like flavonoids, hydroxycinnamic acids, low levels of stilbenes, and some phenolic acids. Depending on the berry, they can be higher or lower. The question we had is that it? There has to be more in berries than what's in these databases. And obviously, it's not it. So how do we work out what's out there in the literature? So we basically conducted a systematic review type search strategy. If you haven't done this before, basically you start with categories and then search terms, and you go to various online databases. We use PubMed and Web of Science in this case. And you get these massive hundreds of thousands of hits, which looks scary, but then when you include your mesh terms, you can get these hits down to a reasonable number of literature to interrogate, essentially. So we had roughly 400 papers at this point, and say 100 of them were duplicates across the databases. So this gives us about 300 papers to go through. You set a bunch of criteria to identify papers that have the data you're looking for. So we were essentially interested in compositional data from berries or from humans who have eaten berries, compositional data from the blood. So when we applied our exclusion criteria, we were able to exclude 174 titles, 41 abstracts, leaving us with 90 papers to extract compositional data from. So what did we see? Well, it's a lot of work. We had 1,260 lines of data from this extraction. Now, I say pre-clean because that doesn't mean we have 100 or 1,000 compounds that are identified. Post-clean, we had about 300 structures identified in these studies. 900 that were excluded were basically synonyms or inaccurate nomenclature or duplicates. And I would say in the polyphenol field, a massive amount of synonyms and inaccurate nomenclature is used. So it makes the cleanup of the database very difficult. So what did we find? Well, we found that obviously there's a lot more than anthocyanins in berries. We have alkaloids, condensed tannins, coumarins, hydrolyzed tannins, lignins, terpene alcohols, and so on. So a berry is quite complex. It's more than just one compound, obviously. So clearly a berry is more than just its flavonoid composition. So that's a good take-home message. But how do we take this data now and establish the contribution of the berry metabolome to the human metabolome. Well, this is our basic workflow which we go through. So we've gone through our target identification and our data extraction, which I've just gone through. We look at various databases for confirmation, such as Phenol Explorer or FoodDB. We look for various mass spec data already out there using databases such as HMDB. But ultimately, we need to get into the lab and spend some money, basically. So we need to purchase all the reference standards we can find for all the compounds which we've identified, either in the berry or in the human. Once we form a reference standard library, then we get to the mass spec. So in our mass spec workflow, everything goes through compound optimization. So basically, we're optimizing for fragmentation pattern, and this is done using direct infusion. This moves on to source optimization, chromatography optimization, and that's kind of the front end. Then we have to get into optimizing the analytical parameters within the mass spec software. So we optimize for various scanning features such as multiple reaction monitoring, and then further going on to scheduled MRM, to advanced scheduled MRM. And there's kind of a break here. This is as far as we can go at this point for the reference standards. Now, we also need to look for compounds which are identified for which there's no reference standards, and that's where it gets tricky. So in order to do this, we have a protocol where we explore pooled clinical samples following berry interventions, essentially. So we can identify these metabolic conjugates for which there's no analytical standards for. Once we've optimized for the fragmentation profiles or the transitions or spectral features within this matrix, then we merge this method and this method to form a final method, and then we can establish precision and accuracy and peak quality through some precision and accuracy measurements. So this is what we basically come up with. So we have a food metabolome database. We've done this for other foods, not just berries, but I'm going to use berries as an example. We've optimized the spectroscopy, so now we have fragmentation profiles and voltage parameters for our mass spec. That goes into a database which allows us then to pick methods depending on our intervention. So if someone wants to do a coffee intervention or a berry intervention, we can pick a method, develop it in silico, test it, validate it, and then apply it to our clinical samples. And also what we need to do is take this historic data from our clinical samples and use that to add back to this reference standard library or this metabolome database to improve on the quality of the data from what we observe in clinical samples. So an example is we use a triple quadrupole MS-MS. So our database is a MS to the N database. So what that means is we have a compound here, if you're not familiar with mass spec, and it has a mass and a precursor transition, say. We optimize for multiple transitions or the masses of its fragments as it fragments, which gives us a fingerprint. Now, why is that important? Well, often in metabolomics, people will use a single transition and a retention time marker. So this is called a single spectral feature with a chromatography marker. And it works fine in simple matrices, but if we have a human matrix with 5,000 spectral features, it's very difficult to have 100% certainty that what we say is there is actually there. So this is, if you were to overlap all the spectral features, you would see something like that. So just to give you kind of a pictorial representation of that, here's a structure here, 3-methoxycinnamic acid 4-sulfate. The sulfate is added by humans, so this is a phase 2 conjugate, and this is a microbial catabolite. So this is one of these hybrid metabolites. In the mass spec, at even low voltages, this sulfate can be cleaved, which makes this peak disappear, and then you only see this peak. So it's very easy to identify the 3-methoxycinnamic acid sulfate as 3-methoxycinnamic acid. Often we won't have retention time markers for things that are biological conjugates. Another example, here's 3 structures here. There's 2 benzoic acids and a cinnamic acid. These 2 fragment to form this structure, And each of these share a common fragment. So it's very easy in mass spectrometry to misidentify one of these structures in a sea of spectral features. So to give you an example of kind of the types of methods that we can pull from our database. So this was a blueberry intervention we were working with a group in the UK. We pulled a method. It had 183 analytes, 111 reference standards, 72 putative metabolites, so these are conjugates which we don't have reference standards for, and it reflected almost 600 qualitative transitions. So these are transitions associated with getting further spectral features from fragmentation. In fact, we've applied this to a number of studies, strawberry, blueberry, various berry-rich diets, and an animal study which we have brain tissue data. So we've used these methods across 1,500 samples feeding berries. You can see just the number of reference standards in each method, the number of transitions, qualitative, and the number of metabolites. So we're identifying anywhere between 120 and 180 metabolites. So this is really quantitative metabolomics as opposed to untargeted metabolomics. So what do we see? We don't see, or we haven't seen yet, any unique biosignatures or biomarkers of intake. Now, when we think of a biomarker, it needs to have predictive power. So if it was to be accepted by medicine or clinicians, it needs to be able to predict a treatment over a control, such as in the case of when you have a heart attack, there's 3 markers which are elevated in the blood, or 4, such as myoglobin and troponin T. And we can use, this has high predictive power, essentially. So we see no clear patterns. And this might be expected based on what you saw about composition of berries and the significant catabolism and human phase 2 metabolism that exists. And it's probably expected based on microbiome and based on what I've shown about the effect of gastric emptying and intestinal motility on kinetics. So this is not something that we would find odd. What we do find, though, is that these flavonoids, which are common in berries, are being broken down into certain classes of metabolites. So we're continuously seeing benzoic acids, phenylacetics, phenylpropanoics, cinnamics, and virilalactones. Now, virilalactones are very low as metabolites as berries, but they are probably our best chance at a biomarker of intake of flavin-3-ols, but probably not for berries in general. Now, interestingly, in these studies, if you look at people that have high abundance of these compounds, we're talking about some individuals, 30 micromolar concentrations of some of these phenolic catabolites. This is quite high. This is higher than hippuric acid in some individuals. So these are things that we shouldn't be ignoring in our metabolomics. So this just shows you all of these classes which I previously mentioned, found in the highest abundance in some studies, reaching 30 to 40 micromolars. Now, interestingly, we've found these compounds in the rat tissue in their brains following the feeding of blueberries. And this actually backs up some earlier evidence that Mario Ferruzzi's group came out with a number of years ago. So why do we see what we see? So as I've mentioned, we got this microbiome diversity, but what about diet diversity? It's believed that our diets are more diverse between individuals than our genomes. So we've got this massive diet diversity and this massive microbiome diversity. Remember, your microbiome is based on every occurrence where you've encountered a bacteria over your lifetime. So everyone collects a different microbiome. So I mentioned that elimination kinetics is going to be highly complex between people. When we do a clinical study, we take a blood sample at a given point in time, say 1 hour. 1 hour gastric emptying is going to be different in one person versus the other, or intestinal transit, or so on. So blood time point analysis is being shown to be highly variable. I've already mentioned the impact on ADME and the impact on catabolism being extremely diverse. So this essentially is what we're seeing as a general picture. Normally in kinetics profiling, we have kind of a model that we have this intestine, It's driving absorption, going to the liver, entering the systemic circulation, and then depending on how it's modeled, it might go through various systemic hepatic passes. Basically, what we're not able to model for is this continuous process as things are moving through our intestine before being excreted, all contributing to these pools, making it a very complicated story. So we're seeing this layered kinetics. So these small increases in peaks here as a result of precursor compounds in the foods are being way overshadowed by these microbial catabolites, where if you totaled them all up, this is kind of what we would see over a 72-hour period. So these things are cumulatively in quite high concentrations. So why no unique signatures? As I've mentioned, absorption, metabolism, and kinetics. And I'd like to spend a few slides focusing on the microbiome because it's such a hot topic in nutrition research and polyphenol research. So this is a phylogenetic diversity tree for the human microbiome as recently published in Nature. So extremely diverse. What we often don't think about is enzymatic diversity. This is the phylogenetic tree for one enzyme in a microbe, or one microbial enzyme. And we know that microbes have a lot of metabolic machinery or enzymes. So we're going to have massive enzymatic diversity within our microbiomes. So if we kind of follow down that route of a metabolic phylogeny hypothesis, I guess, what can we learn or infer from microbial evolution that will help us get a handle on what's happening with our polyphenol metabolism? Well, microbes are catabolic in nature, and they evolved to break down plants, obviously. The biosphere initially only had plants basically in it for the microbes to consume. So they developed pathways to consume fibers and lignins. These are primary plant metabolites. So if we consider lignin as a very complex polymer, this is just a hypothetical example, but all of these units are found in a lignin. It's polymeric makeup. It has various polymers in it like resorcinol, phenylcoumarin, biphenyl, and if you note, these are all polycyclic aromatic hydrocarbons, and lo and behold, that's the same thing flavonoids are. So I'm going to assume that flavonoids are being metabolized using similar or the same enzymes as lignins, and the evolution, or at least the progression of what the microbiology has learned about lignin metabolism in the past decade is quite substantial. It just hasn't yet been applied to polyphenols. So these compounds are broken down to something called biaryls, and these metabolic enzymes are highly characterized in bacteria. So these are the types of structures. This is just two of them, GGE and HMPPD, but they look very similar to polyphenols. Especially anthocyanins at neutral pH. So if we consider that the enzymes to cleave these are highly established, we haven't tested these enzymes for polyphenols, but it's reasonable to presume that they would cleave polyphenols in much the same manner. So what we have is this kind of catabolic funneling from these polymers to these biphenyls to phenolics. And lo and behold, two of the key phenolics in this lignin metabolism are gallic acid and protocatechusic acid. These are both reported continuously in studies feeding high polyphenol interventions. The funneling goes further on from there into our ring fission products, things of this nature, OMA and 5-CHMS. And these are compounds that we're not even really looking for in the polyphenol field. So we've got a long way to go in establishing the total catabolic pathway for polyphenols. But basically, the question we need to ask ourselves is, could plant metabolites, such as secondary plant metabolites like polyphenols, be susceptible to the same metabolic machinery as primary plant metabolites? And it makes sense to me to go down that route of hypothesis. Now that begs to differ and means that we need to consider a lot more confounders in our human interventions, because what I've showed you is there's multiple substrates producing the same metabolites. And if we think of things like chlorogenic acid in our diets, coffee, potato, but also things that we don't often consider, like ellagic acid or some of these larger polymers or ellagic tannins. We're unable to wash these out of the system. So when we conduct human interventions or epidemiological studies, do we really know what's in the background diet and what's causing the effects that we're seeing? Going back to 2 slides I took from one of Mario's slide pools, thank you, Mario. We need to consider these because they're in high abundance. We have polymers, monomers, and our phenolics. We're consuming grams a day. How are they contributing to the metabolome, and how is this impacting health? So my talk, I said, was about risks. So this is where we get to the risks portion. We need to consider diet variability between individuals. People in different households have completely different diets. And we need to consider this microbial enzymatic diversity, which we tend not to because it's not likely to be captured by 16S RNA microbial community diversity screening. So we need more studies using shotgun sequencing to be able to get a handle at what is the metabolic diversity of our microbiome. So as I mentioned, microbiology as a field, aside from polyphenols, has done a lot of work in the past decade or two to characterize the metabolism of lignins in soils or various other substrates, basically. So there's a pathway called the epoxybenzoyl-CoA pathway that's been characterized for benzoic acid catabolism. There's oxic and anoxic pathways for extradiol and intradiol cleavage, which just basically means either between the hydroxyls or on the other side of them. These are established again for benzoic acids. So there's multiple ways for microbes to break open benzoic acids, and that's why it funnels things towards those structures. There's gallic acid metabolic pathways which have been characterized, and I've told you that gallic acid is a common metabolite of polyphenols. Essentially, the microbe's goal is to open the ring, produce oxalic acid and pyruvic acid to feed their energy metabolism cycle. Protocatacoustic acid, there's metabolic pathways and enzymes already established for that. Basically, the microbes take hydroxybenzoic acid, add another hydroxyl so it can break open the ring. Even our phenylacetics and our cinnamics, which we're seeing in all our polyphenol interventions, the catabolic pathways for these are already established in microbiology. And lastly, hydroxycinnamics, it's the same. We have things like ferulic acid degradation pathways, which are characterized, which go through a number of intermediates, including vanillin and vanillic acid and protocatacoustic acid. So again, we haven't established these pathways by feeding pure polyphenols to microbes and characterizing the enzyme that's breaking them down, but it's been done in the soil microbiology, in industrial processing, in biofuels. So a lot of these pathways are well characterized. So it's a complicated story. How do we move forward? Well, I want to talk about managing risks associated with mass spectrometry, and there's a lot of people that use mass spectrometry in this room, and there's a lot of people that don't. So I've broken it down into some fun little analogies, which I'll take you through. But essentially, we need to manage risks in metabolomics. We need to prevent false positive identification of structures because this causes researchers to chase hypotheses, and they could be chasing the wrong metabolite down the rabbit hole for considerable periods of time, years, maybe decades, when they're exploring mechanisms of action or the health implications of an intervention. So this is a significant concern. So often we use retention time as a single chromatography feature, which is good in a simple matrix, but when you're talking about metabolomics where you could have 5,000 signals, we need to consider we have structural and stereoisomers, artifacts, and adducts, and I'll explain a bit about what those might be, but this makes it problematic sometimes just to use retention time as a marker. So let's go into my analogy for those of you that don't follow mass spectrometry. So we talked about retention time. So time is one distinguishing factor. This is kind of an analogy that I took from Susan Sumner, and I've termed it, have you seen Dave? So this is Lego Man Dave. He's 4 centimeters tall and 1.6 centimeters wide. if you had to describe him, this is what you would describe, but this is his fragmentation pattern. So similar in the mass spec how a molecule fragments. If we hit Dave with enough voltage, he's going to fragment into these parts. Dave can wear many hats. These are biological conjugates, so glucuronides, sulfite, glycine. So these are all spectral features that we can use to identify Dave. If we know at what voltage Dave fragments at and under what environments, we have more predictive power in identifying Dave. So what are we up against in metabolomics? This is literally what we're up against. So where is Dave? Like, maybe that guy's Dave, maybe he's Dave. I'd say we need more data. We can't just use one feature to identify Dave. Now, what if Dave is conjugated? So now it gets even harder. Dave can wear many hats. So again, this is what we're up against. He can also be an artifact or an adduct, and I'll go into that a bit later. So we know what Dave's made up of, and we know how he fragments. Picture this brick wall as our baseline or our background noise within mass spectrometry. So it's complicated by the number of signals. So we need more data to differentiate Dave, but we have various scanning techniques that we can use to identify him with more certainty. So it's also complicated by many common signals in our background matrix. So we might see Dave's at multiple periods of time, and you'll see more of Dave the closer you get down to your baseline noise, basically. So we need to know our background matrix and how it affects our spectroscopy. So it's complicated by isomers, or things that have similar mass. So we might see multiple Daves when we're only expecting to see one of them. Again, more data is required. If we turn this on its axis and we are going to look down the time axis, we could see multiple masses at similar retention times. So this complicates things. We need to further interrogate our data. And it's complicated, as I mentioned, by conjugation. So Dave can wear many hats. Now, to get these hats off, we have various techniques. We can use various scanning procedures. We can use various techniques to identify one Dave from the next, essentially. One of the main ones is voltage ramping, but we can also use a different platform that maybe has a higher resolution that can give us more data about Dave, or break down this brick wall a bit more so we can see more of Dave. So I mentioned voltage ramping. This is all things that all mass spec can do. You can play around with the voltages. What does that do? Well, if we know the voltage that fragments Dave, and we hit Dave with a lower voltage and his head falls off, we know it's not Dave, basically. He's fragmented under a different condition than we're expecting. Same with if we want to knock Dave's hat off. We know the voltage required to knock his hat off. If it falls off at a lower voltage, it's most likely not Dave. So we can conduct voltage ramping experiments to narrow down who Dave might be. Remember, this is his fragmentation profile. Now, it gets a little more complicated than that because we have things called artifacts within the mass spec environment. So these are things that might not necessarily be in your sample, but they're produced in the gas phase as a result of high voltage, basically. So these are things like water elimination, hydration, hydrogen elimination, radical fragmentation, and so on. These can affect the mass of a structure by as little as plus or minus 2 units to as high as plus or minus 17. That means that one structure can be seen under a lot of different masses. So that means Dave can have different heads that aren't really real. They only exist in the mass spec for short periods of time, basically. So to give you an example of that, for our polyphenols, and it seems to be a little worse for the anthocyanins, but it can happen for any of the polyphenols, as a result of this cyclic ring and the electron localization, essentially, and this secondary C ring, which is not a true benzene ring, these structures are prone to a lot of spectral artifacts, basically. So we can see this structure at a mass of plus or minus 1 based on certain rearrangements. We can see it with a mass of plus or minus 2, plus or minus 14 or 15, plus or minus 16, and as you would expect from my previous slide, plus or minus 17. These are documented throughout the flavonoid literature and the polyphenol literature, these artifacts. Now, you might think, well, maybe these are just artifacts found at really low levels. We don't need to consider them. This is the artifact peak here. These are seen at e to the 8 often, which is 1 with 6 and 8 following zeros. So these are found. You can produce these at very high abundance, much higher often than your signal that you're actually looking for. And in fact, I just showed you for cyanidin, but just to let you know, we've looked at a bunch of other anthocyanins, and this just shows us that we can produce these artifacts from -1 to +17, and here's the collision energies where we see the maximum intensity. We often find them at 10, a collision energy of 10, which is low ionization efficiency or low ionization voltage, which is most commonly used in metabolomics. So something to be concerned with. So what that means is, I'm not going to go through the whole table, but just this first line, cyanidin O-glucuronide can have the same mass to 4 decimal places as pelargonidin O-glucuronide. It can also have the same mass as these native anthocyanins as found in plants. So this just means that the spectroscopy is complicated, and we just need to make sure that we properly establish methods and interrogate our data. And the same can be said for these structures here. We can see basically Dave with many masks, if you want to put it that way. So we need to manage these analytical risks to prevent false positive identification, inaccurate quantitation, and a lot of this just comes from a lack of understanding of the background matrix and how to set our mass spec to avoid these issues. So to give you a few examples with some spectrum, why capture just one transition? Well, maybe we can get away with that if we have accurate mass, but we don't often have accurate mass in metabolomics because the cycle time is quite low. So we often have triple, quadruple, and nominal mass, which often requires we create more spectral features by fragmenting the structure. So what that means is we have Dave's fragmentation pattern. We want to be able to see that this is Dave and this is Dave's makeup, basically, as we know it under conditions that we've optimized for. Here's another example here. How do we optimize for a compound we don't have a reference standard for? So in metabolomics, we don't have reference standards for everything, particularly biological conjugates. But if we have the unconjugated structure, so say 2-hydroxyphenylpropanoic acid, we can optimize for that and then optimize for the conjugate, and we should be able to see the fragmentation with or without. The problem is if we don't optimize this sulfate group here, which makes up this transition, at low ionization efficiency, often is cleaved in source or before you get to your detector. So it's very easy. This peak would disappear and you would only see this peak and you would misidentify this compound when it's actually this compound. So going back to Dave, we know what Dave's made up of. We optimize for him and then we optimize for his conjugate, and we should be able to put the pieces back together to form conjugated Dave. We also need to interrogate our spectra. So the mass spec will do whatever you ask it to, if you're, not everything, but it will look for a compound if you tell it to look for a compound. And it will spit out an area and intensity. If you do no optimization, if you just take that area and intensity and establish either quantitation or establish nominal difference, there's a lot of danger. You need to look at the raw data when it comes to mass spec, or look at the signal. So when is a peak? Well, that generally requires some idea of your background matrix and threshold manipulation. So essentially your threshold should be optimized so your software does not interrogate noise. You need to establish this in a matched matrix relative to blanks across a range of concentrations and across multiple replicates. Now, why all this work? Basically, well, here's 3 injections of a blank and 1 injection of a low concentration analyte. The mass spec it's integrated basically and generated an area for you, but you can see the noise within this time window is 40, and it's integrated below 40, which is quite dangerous, and I'll show you why in another slide. What you want to see is a really nice integration above your background noise threshold, which you can only establish in a match matrix using multiple blanks, basically. So basically, if we go back to Dave, here's our baseline. We've set this blue line as our threshold. This is Dave just peaking up above the threshold. Is Dave sufficiently above your baseline for you to have some accuracy in your quantitation? I would say no. So when is it good enough, basically? Well, often what people do is they set their thresholds too low to improve their limited detection, which is dangerous because it leads to false positive identification and inaccurate quantitation. So that's why people often set their signal to noise at 3, but theoretically it could be 4 or 5 depending on your background matrix. As I've mentioned, lowering this is dangerous to improve your LOD, and manual integration is dangerous. So here's another example. Here's a compound at 1 concentration injected a number of times. You can see that the mass spec has integrated it differently each time, and that's because the threshold has been set too low. Like it's integrating at 10 when across this time window we're seeing probably 50 would be more appropriate. So we might have to increase our limited detection to get better integration and more accurate data. Again, the same as before. How much of Dave's head can we quantitate with accuracy? That much. We want to see the whole head, basically. Same with peak or noise, a game I like to play. A lot of people will say, look at these great peaks. They're very thin. They're actually not peaks. They're just random signals picked up in the mass spec. And I would say this is just noise also. For one, it's got a 6-minute time window, and for others, it's very jumpy. So this is just picking up random signals. The mass spec has integrated it and provided an area because we've told it to look for a peak at that time point, and we've set our threshold too low. Again, this is dangerous. So do we have enough data points to accurately establish Dave's true abundance? We need a clear picture, basically. And if you're just looking at your area data, you'll never know. The same is when we optimize our reference standards in our matrix, or if we're looking at a putative compound that we don't have a standard for, it should stand up to a dilution effect, meaning if you dilute your sample 3 times, it should be linear within your matrix. And often that's not the case, and that tells us that we've probably identified the wrong structure. So back to our analogy, does Dave get smaller the further away from him you move? Logic dictates that yes, and a dilution, that you should see less. So risk summary here when it comes to mass spec is, as there are a few reference standards available to confirm optimization, particularly of polyphenol metabolites, false positive identification is likely. So without ample experience, method development, validation, and most importantly, interrogation of your data, metabolites will continue to be falsely identified undoubtedly, and this will hinder future discovery, as I mentioned previously. So the take-home message is mass specs have become cheaper, everyone can buy one, everyone can use one, but if it's a black box to someone, we need to treat it with respect, question it, but also fear and respect it. Demystify it by making observations, by looking and interrogating data. So how do we wrap this up in a take-home public health message for polyphenols, as that's why we're all here in the Plants for Human Health Institute? Well, I think considering the structural similarity of the polyphenol metabolites, as I've shown you, derived from precursor phytochemicals, so essentially which are high in our diets in a diversity of foods, such as polyphenol-rich fruits and vegetables, processed products such as coffees, teas, cocoa. All of these compounds and foods likely contribute to the observed health effects reported in the epidemiological studies when we look at increased fruit and vegetable consumption, or increased polyphenol tea consumption or cocoa consumption. And the trick really to moving forward is we just need to simply manage the risks, particularly in respect to metabolomics, develop and utilize high quality data which will allow us to produce sound hypothesis, and this will drive future discovery. So I'd like to thank you for your attention, and that's all I had.